Good morning, everyone. Today we will be seeing NMR spectroscopy and how we are going to get the structural information from NMR spectroscopy data. And we discussed about like 1D NMR spectroscopy, how we are going to do, how we are going to see the spectra, what would be the spectra pattern like, and what would be the spin-spin coupling, what would be the chemical shift positioning, what are the factors which are affecting or influencing the chemical shift positioning with respect to the reference what you are using it. So if you recall all this together, then what today is, we will be going from 1D to 2D NMR. So today's our focus will be more on a 2D NMR. And what is a 2D NMR? How we are going to understand the spectra of a 2D NMR? What is the relation between 1D and 2D NMR? And finally, how we are going to get the final structure from different experiments of 2D NMR. Okay. Now, this is one slide which I wanted to show you. If you see here, this is basically how the spectroscopic techniques have evolved over a period of time. And in why I wanted to show this is because if you see here, this is the first in 1923 the organic X-ray was used, right, as we saw in the previous X-ray crystallography session. And from there people started exploring different methods. And if you see, the first analytical use of NMR was done in 1955, and for like as the time evolved, the requirement also evolved, and basically they all in the later stage they have all settled down to 2D NMR. So this is COSY and NOESY, and this COSY and NOESY are definitely 2D NMR techniques. So this how today I'll be focusing on this 2D NMR techniques. But why I showed you is, as we move ahead down the line, people have understood the importance of NMR, and then the NMR technologies have also evolved. So NMR is not only a simple system where you just get one spectra and you try to interpret that data and get everything, but yes, at the same time from NMR data you get a lot of informations. If you try to cross correlate with different types of spectra, you will get enough information to give you the structural elucidation, in contrast to other techniques where there are a lot of like if it may not be that so many experiment wise, it is not divided, but at the same time you get a half-baked informations. So in this case we have to do number of experiments. Different experiments will give you different informations, and when you try to pull all the data together, then only you get the final structure without any limitations. This is the spectra in 1D platform, or a 1D you can say a proton NMR. And if you see the spectra, they have lots of peaks. Some peaks are resolved, some peaks may not be resolved because there may be overlaps. And if you zoom in, or if you can see, there are a lot of peaks which are lying shoulder to each other. Okay. Now, in spite this of so many crowds of 1D NMR, it is very difficult to decipher exactly if it is not if it is a pure structure, then also it may be difficult if it is a complex one, and if it is not a pure substance which you're trying to identify with it, then it will be much more complicated. So just to give you a understanding, this is basically at 0 ppm. At 0 ppm you have the reference peak. So this reference peak is the reference solvent which you are using it. It can be tetramethylsilane, that is a silane which has 3 or 4 methyl groups, right? And all these methyl groups, so 4 methyl groups together, all the protons will give you only one peak. So we adjust this one peak to the 0 ppm position in the chemical shift scale, and then rest of the scale or the rest of the spectra will get aligned based on its respective chemical shift positioning. Now These are some of the identified one, and from this identified one you can see these are already known because we have a library. From the library we can find out if this is the ppm position, this is the J coupling, this is the spin coupling, it can be a doublet, triplet, what is the multiplicity of that peak, and based on all these informations, if the values matches with the library, you can annotate them as it is annotated here. And interestingly, you see a residual water peak. So you see here, this is a residual water peak, and this peak is looking different because it is going this way. So what happens when you have a water, the water is full of protons, right, H2O, and this protons will give you very huge signals. So what happens is there is a opposite pulse given at the same resonance at the same ppm position, so that the signal at this position is submerged or it is subsided. Now once this water signal is subsided, what will happen, the effect of having one huge peak is it will definitely make the other small peaks go away. Because if you have a huge peak here, which is like almost like 10 folds, 20 folds high compared to the average of the peaks, so what will happen is this small peaks which you are able to see now, they will not be prominent enough because they will be submerged, they will be subsided. So this one small peaks, how you have to understand or how you have to get these peaks, because we know this is a water peak, this is a residual peak. Now this residual water is the water where your sample is absorbing either from the air in terms of humidity, or by mistake if you prepare your sample, you dilute your sample into a water base, right? So to avoid this situation, what usually one does is you use, instead of water, normal water, use D2O. Now this D2O will not have resonance, it is not magnetically active, and it will not give any resonance in the NMR presence, right? So what happens is, in presence of a D2O, you will not get any signal here, and you dissolve in the D2O your sample, whatever, if it is a pure compound, protein or whatever. Now the problem is, if you have to also understand, see, D2O will dissolve only which are polar compounds. So if you have something with alcohol based, something with OH, something with aldehydes, then they will get dissolved in D2O. But if you have lipids, fats, right, also at times oligosaccharides, complex oligosaccharides, at those cases what you have to use it, you need to use a solvent in which it gets dissolved. So there are a lot of solvents which are available, you have to just use them, and based on, first decide the choice of your solvent on which you're deciding based on the sample what you're going to study. Okay, so this, if you blow it up, you see there are a lot of, this is like a forest of peaks. You see here there are forest of peaks, and it makes the life of a person very, very difficult in understanding what these peaks can annotate. It is very easy if you have individual peaks like this which are prominent, and these are the peaks, they go for this, and it is easy for you to annotate these peaks. But the life is not that easy, because when you see something like this, so you have all this, now how you are going to solve it? Then 1D NMR would not be enough. Okay, so you did a 1D proton, you did a 1D carbon, you can do 1D phosphorus, nitrogen, whatever, you will get all this spectras, and then you have to move ahead. So there are databases. These databases will give you a lot of information, because people have worked on this databases. There are a lot of repository which are available, and this repository, from this repository what you can get the library, and also the information, the splitting, the chemical shift positioning. So you can use these libraries. So this is called a predictive method. where you do where you understand the relative and absolute stereochemistry of your sample. So for example, if I talk of a glucose molecule, so this glucose will have a D as well as L rotatory, right? Dextro and levo rotatory. So one will be clockwise, one will be anti-clockwise. So when you are looking for rotations, so this rotations can also give multiple peaks, because they are resonating at different position because of their rotations. So rotation is something which is like inherent property of for example a glucose, but because of this rotation patterns, you are getting different peaks at different point of time. So ideally, if you are, suppose you are having C6H12, in this case, if you are saying ki H12 protons are there and I'm expecting this, it may not be always true, because you have to also understand the stereochemistry of the chemical, of the compound, because they may also keep rotating. There can be a cis trans rotation, there can be a DL rotations, there can be aromatic rings. So all these things you have to keep in your mind that this can happen with your sample, and then based on that the 1D spectra can run, and it can look very different from what you are expecting. Okay, now second is you are comparing the chemical shift values. Yes, you have to compare every time, and the structural properties of a compound are inherent to the specific, as I said, to the stereochemical arrangements of small substitutions on its carbon backbone, and it can be also independent from the rest of the molecule. So these two criteria, it can be either independent or it can be dependent on the stereochemical arrangements or rearrangements, you can say. Okay, so these are the small informations which will help you to get a final what is the understanding of your structure. But 1D alone will not help you to get through this. So what we do is we do a 2D NMR spectra. Now here you see this is 2 dimension, 1 dimension and the other dimension, and these 2 dimensions are having single 1D spectra. So this is 1D spectra, this is 1D spectra. When you correlate the data of these 2 1D spectras, you get patterns like this. Okay, now you see here, for simple understanding, you trace. So what you do is you draw a straight line on this, and you draw a straight line from these peaks. The intersections will, you see, from the intersections you get a peaks, or you get some dots. Okay, now these dots, depending on the splitting, you can have single dot, you can have doublet, you can have 2 dots, triplet 3 dots, and quartet you can have 4 dots, right? So depending on the splitting, spin splitting, you can have number of dots here. But these dots or these parts are not so no, just to get a correlation, but you can do a cross correlation. So like if I want to do proton correlation, yes, I can do proton correlation, just to be assured that whether whatever spectra I got in 1D NMR, whether in the 2D they tell you the same message or not. That is first thing. Secondly, see, when a structure is there, you have a carbon backbone and you have protons onto it. You can have a functional groups, you can have oxygen, nitrogen. So what is important is, if you can find out the basic backbone and correlate with the neighboring protons, then the most of the structure, or the 90% of the structural elucidation is done at this point. So we can do not only proton correlations, we can do proton carbon correlations, and proton carbon correlations have much more information compared to a proton, right? Because you see the carbon 1, carbon 2, carbon 3 all will be associated with different types of protons, and this proton and carbon, when they're correlated, you get a informations of their distribution in the carbon. Now you can also do carbon proton and carbon nitrogen, so that you get the entire understanding of your structure. And there are several experiments to carry out that, right? So it is not as simple like if you just put 2 spectra, 1D spectra, and just draw it. No, it is not like that. There are several experiments, and I'll discuss about those experiments in details in a while. So they can be correlations, if you want to do COSY, COSY, NOESY. So they are different experiments, and through different experiments we're trying to understand different aspects or information from your sample. Like for just outline, if you are doing a COSY, it will be through the bond length. So in the nearest 3 bond length, it can tell you what are the neighboring protons or carbon are there, and you can do a correlation to 3 bond length distance. Now if you want to do COSY, then you can also do not only through bond lengths, you can also do through the spatial orientation. Like through space, they are not connected through bonds, but they are structured in such a way that through space they are very close to each other, and that understanding you can do it with the extension of NOESY and COSY. Okay, so to name some of them techniques is a correlation spectroscopy. As I said, correlation spectroscopy is through the bond length. So you can have different bond lengths, and through the bond length you can find the correlation to nearest 3 bond length distance. Okay, now if you do different like heteronuclear, then we have a Hector. We have heteronuclear correlation spectroscopy, its short form is Hector. Then it is HMQC, that is again heteronuclear multi-quantum coherence. Okay, we have NOESY. In NOESY we have nuclear Overhauser effect. So Overhauser effect is something where we are not only getting the bond informations through the bond length, but we are also getting the spatial orientation in the close vicinity. So that is through the NOESY. Then inadequate, yes, incredible natural abundance double quantum transfer experiment, where we have the natural abundance is there, but we double the quantum transfer and then we get the information. And there are several others experiments like this. So COSY, correlation spectroscopy, it's a proton-proton. It can be cross correlation also, but usually it is done with the same, like with the proton-proton correlation spectroscopy, and is a useful method. Is a very useful method, which for determining which signal arise from the neighboring protons. Right. So at times, like usually when I used to do it, we faced like 3 bond length is very good. We can extend to 4 bond length also, depending on how simple or how complex is your molecule, right? So we have to have informations in the bond length. Then COSY is the first step which you can do it, and you can see the correlations of the neighboring protons in the bond length, right? Next, if you want to do it a step further, then we go for a COSY. Now, as the name says, total correlation spectroscopy. Okay, so in total correlation spectroscopy, as it defines, uses a spin lock for coherence transfer. During the spin lock, all protons of a coupled system become strongly coupled, leading to cross peaks between all resonances of a coupled system. Now, what is happening is, when you are doing a total correlation, what is happening is, it is through the bond length, and it is the total correlation with all the protons or all the carbons, whatever probes. So if you are doing a COSY with a cross correlation, if you are doing a COSY with the proton-proton correlation, whatever you are doing it, but it will give you, suppose you have one proton, it will give you information of all the correlations with all the protons in the neighbor, not limited to 3 or 4 bond lengths. So first is you are limited to 3 or 4 bond lengths, and the third, the COSY is you are correlating with all the neighboring or all the protons in that particular structure, right? Then comes heteronuclear. Again, heteronuclear, I think the most important thing is when you have both carbon as well as proton, and you are doing a heteronuclear correlation, usually between, yes, as I said, a proton and a C13 resonances, and it is mediated by J resonances, that is a spin-spin coupling for carbon and nitrogen, carbon and proton. So this is for heteronuclear cases. And yes, Overhauser effect. So again, it is a very important experiment in a 2D NMR, where spectroscopy method is used to define or identify nuclear spins undergoing cross correlation. to measuring the rate of cross correlations. Okay, so cross correlations, like it is essentially a cross correlation experiment where proton and carbon and nitrogen and proton, so there has to be two different what do you say, probes you're using, two different types of probes you're using to get from two different heteronuclear resonances, and then you are seeing the cross correlation between them. Okay, so cross correlation, to make it simple, is when it is nearby and when it is for a different nuclei spin, we can have a cross correlation. Okay, so different nuclear spins identify the cross correlation or the cross interactions between them, then we are using a NOESY as an experiment. And again, a step forward is heteronuclear Overhauser effect spectroscopy. So correlations between protons and heteronuclei that are close in space. Now here in HOESY, so we are doing it not only through the bond length, we are also doing it through the spatial orientation. So suppose two molecules, they are closely positioned in space, but they do not have a connection through a bond length, but they are spatially very close to each other. So those informations can also be collected using the HOESY experiment. Okay, so again, a COSY spectra. So if you see a COSY spectra, this spectra, if you can understand the small parts here, if you see, this is, you have to draw, what you have to draw is, you have to draw lines, straight lines from all the peaks. So this is one peak, this is a triplet, this is a triplet. You draw all the lines, and you draw all the lines from these peaks where they will, the cross section, the cross section position will be a peak, and you have to match whether this particular cross sectional does match with all the peaks on the other side. So you have to match the peaks. If the peaks matches absolutely, then your identification, or it's a doublet or triplet, whatever you have identified, it becomes a confirmatory study. Okay, so this is a correlation spectroscopy, commonly used for regiochemical assignment. So like you have a different regions, and for a regiochemical assignment we can use it. The cross peaks appear if spin coupling is present. So this is important, because this cross peaks will only appear if there is a spin coupling. It may also be position, it may also happen that there are two peaks. Okay, they look like a doublet, but they are two single peaks. We have assigned them as doublet. So when you try to cross correlate them, these cross peaks, they will not give you the cross peaks for both the dimensions, or for both the single peaks or 1D spectra. Why? Because these two are not a correlated peak, they do not have a spin coupling, but they are two single peaks which we have wrongly identified as a doublet. So the second important thing is, the wrong identification of multiplicity can also be done away with when you are going for a 2D. Now protons that are separated to 2 or 3 bond length can also be identified. Yes. Okay. Coming to Overhauser effect. So Overhauser effect is what? Overhauser effect is, like if you see here, to be very, just a rough sketch here. This is one proton, this is another proton, and these two protons are connected through a very complicated bond length. This bond length is not even close to 3 bond length distance, not even 4 bond length distance, not like that. Okay, but the interesting fact is, suppose this is one proton and this is another proton, they are sharing a very close vicinity in the spatial distribution or spatial orientation. So when they are closely positioned in a spatial orientation, they will influence each other. Although they are not connected with a bond length, but they will have an influence on each other. That is bound to happen. But we are missing when we are doing a 1D, when we are doing even simple 2D correlations, then also we are missing. So to find out this information, one of the easiest thing is to do a Overhauser, or study a Overhauser effect. Now, when we are doing a Overhauser effect, what we do, we have a common like a NOESY or HOSY experiments we can run, and the local field at one nucleus is, you can see here, is affected by the presence of another nucleus. The resultant is a mutual modulation of resonant frequency. So if you this you see, these are like two magnets. Suppose two magnets or two dipoles, if you bring them close together in space, although they are not connected, but still their magnetic field will try to interact with each other. And similar thing happens when in a conformation, when spatially two protons are positioned very close to each other. So to identify, to get information on this aspect, Overhauser effect is a very useful experiment. You can get the informations that not through bond length but in the spatial orientation, which are the neighboring protons or neighboring nucleus that is actually affecting the entire spinning of my currently studied nucleus. Okay. One quick or a small example of a propyl benzoate. You see here, this is a NOESY spectra, and in this NOESY spectra what you're doing is we are trying to correlate. So you see you are doing the cross peaks. So what you see is this is basically, these are we say as cross peaks. Okay. So this cross peaks because you are correlating two 1D spectra into this, and you are trying to get the structure here. You are trying to get the structure here, and in this case you see the cross peaks, they should match. So they should match not only with individual peaks here, but they should also match with the cross peaks here. And when they match, what will happen? It will show correlation to protons that are nearby in space to about 4 angstrom distance. So spatial distance, if it is near to 4 angstrom distance, or at times it can be little bit more than that also, but depending on what is the magnetic field, external magnetic field you are using it, so you can do that. You can extend, or you can identify, or you can the distance can be little bit more also, but the idea is the neighboring in through space, you have to identify the correlations, and it can be done here. Interpret by noting which protons are close to one another. Very useful for stereochemical analysis. For stereochemical analysis, this is very important, because if you are using or if you are studying something which is like, for example, if you are using glucose, then also the D and L changes. There can be also chemicals where a cis trans changes are also present. So this stereochemistry understanding, or this information, can also be understood very well in this NOESY or Overhauser experiments in general. Okay. Great. So protons that are we say geminal or vicinal will still appear because they are close. Now again, so this is the stereochemistry terminology I'm using it. So the protons which are positioned in a geminal position or the vicinal position, depending on their orientation, they can also be studied. And finally, when you cross correlate with both the spectra, it can be with proton, it can be proton carbon, and when you do that, you get the cross peaks, and you finally understand what is my structural backbone. That is important. When you do 2D, you get an information which is much more solid, which is much more robust, and it is much more confirmatory compared to a 1D data. Okay, so now let's make things a little bit more complex. So in this, you see this is a steroid, and this is a particular steroid, and we're trying to do it for a, so when you're doing it for a complex structure, you get peaks like this. So now this is what you see is one spectra, and this is another spectra, and this is the cross peaks, and these are the peaks which you are matching it. So what happens is, when you try to correlate, you have to match not only this peaks, but this peaks should also match with the cross peaks here. So this is a diagonal peaks, you can see here, almost like a line, and it should match with both. It is not, if you draw a straight line from here, it should match the peak here and it should also match with the peaks here. And that is where, suppose one compound, it is bound to have multiple peaks at multiple ppm or chemical shift positions. Now if all the positions are matching or not, so in a 2D we say, okay, I see one peak here, I see one peak here, I see one peak here, and we say from the library, okay, this is the positions, one particular compound, this is going to have 3 peak positions with a multiplicity so and so, and with a J value this, and you match it and you say happily. But that may not be always true, because of the other factors, the stereochemistry, because of the influencing, like Overhauser effect, as I was telling. So when you do a 2D, what happens is, when suppose I have one peak position here and the same compound also gives a peak position here, so what will happen is, when I try to correlate, it should correlate with all the peaks. So all the peaks of a particular compound will correlate with all the peaks in the cross peak as well as with the other peaks in the heteronuclear dimension. So if it is another spectra, it should match with all the peaks, and then only we can confirm, okay, these are cross peaks, and then only this is the structure which I am getting it. Okay, so this is something which you need to understand, and it has to be understood really well, because when you like the common practice is, we used to take a printout of this 2D spectra, take a pencil and scale bar, and with this draw lines. And when you make all these grids or drawing lines, all the lines from this peaks, on the lines from this peaks, and you see the junctions where they're matching with this cross peaks or not. When all the cross peaks will match, then only you know this is my, actually I have assigned, or I have understood how to assign this, or whatever the spectra is telling and whatever I'm deciphering goes hand in hand. Otherwise you will be simply saying something, actually it may not happen. Okay, so this is another a simple 1D experiment, series of 1D proton experiments of some of the natural, you can say, products which are available in our day-to-day life. So protein is one of them. So you see steroids, right? So steroids, then other things like terpenes, carbohydrates, peptides, alkaloids. Now this all are there, and this is one simple protons are there, and CDCl3 is used here. Okay, so CDCl3 will also is used as a solvent here, and you are not getting a peak from CDCl3, that is why. Okay, you are not I'm not showing it here, that is why. Now the point is, you see here the distribution in 1D, it looks like very, okay, I identify this one, identify this one, identify this one, and all three are present. Okay, this is like an alkaloid. Okay, so for a peptide, I know this peaks, I know this peaks, and it is a peptide. Steroid, I have one peak here. Marker, I have this peaks here, I have one peak here, do it. But absolutely, in case of a carbohydrate, simple, you cannot do that. In case of steroid also, you cannot say it's 1D proton just like this. So what we have to do is, we have to do a combination of 1D as well as 2D experiments, get all the peaks together, get all the data pulled together, and finally come up with the structure. Okay, so there are methods of doing it. So one simple thing, you can also quantitate. Suppose I have used carbohydrate, as in this case. So this is a carbohydrate, I got a 1D NMR of this, a carbohydrate spectra, and from the carbohydrate spectra, suppose I used 1 millimolar concentration of dry carbohydrate, any carbohydrate it can be, I have selected, right, and I want to quantitate it from the NMR spectra. So what you can do is, you can integrate the peaks. So the quantitation is can be done by integrating the peaks. So whatever peaks are there, if you integrate, or the area under the peak, to be very simple, if you can find that value, that will give you an estimate of the concentration of the particular compound, or what your study, like may be a pure protein, may be a steroid, may be a terpene, whatever it is. So all this you can quantitate. Now why I'm saying this, because apart from structural elucidation, you also get an information about the quantity. Suppose I have extracted some pure compound and I want to quantitate it. I need not take any other technique, or I need not go to do any other method. I should not be going for any other method to quantitate it. I can do it with the NMR. I can absolutely do it with the NMR with a very you can say accuracy, with a very high accuracy. Okay, so what you do is, you see here, this is a 1D NMR, and the integration of all the peaks of a particular, so this is this is again a sugar residues. So from the sugar residues, you see here, you get the peaks and you integrate the peaks from the proton, from the carbon, and you get the number of sugar residues which are available. Now when you are going to do it for all, like so suppose you want to do it for a constituent monosaccharides, so what you have to do is, you have to have all these experiments. You need to do a homonuclear as well as a heteronuclear experiments. In homonuclear experiments, what you're doing is, you are doing, apart from like a 1D experiment, this is the 2D experiment. You can do correlation, you can do total correlations, you can do Overhauser effect. Okay, so 1D 2D toxes can be done absolutely, and these are all for the proton. So why it is homonuclear? Because also this is a 2D experiment, but you are doing it for proton only. We can do then comes the heteronuclear, yes, where you are doing is HMQC, HSQC, okay, and correlated with the toxi. And this data, along with the previous data, you are putting down in the form of a table. So we are in the table, we know the chemical shift that is given in delta, that is for the proton. We know the chemical shift of carbon, that is again you can see here in C13, and then comes the final J value of the spin-spin coupling. So the spin-spin coupling will only happen with the protons, it will not happen with the carbon, because carbon is the backbone. You will have only the side chain of the protons, and they will have the spin-spin coupling. So matching the J values, once you do all this rigorously, then you can come up with the final configuration. So as you see here, in case of a anomeric configuration, what you do is, you need the GA value is very important. You need to have the ppm positions for proton as well as carbon, and then you can do it. Similarly, linkage and sequence. So you see here, this is a Overhauser effect. So with the Overhauser effect, which is less than 3 Armstrong here, you can identify that also with the spatially, whether they are oriented and how closely they are positioned. You are also having it, and then you can also do it for a ring. So if it is a ring, if it is an aromatic structure, then their J value will be different. They will have a de-shielding, which we discussed, right? So they will have a de-shielding effect because of the aromatic ring, and the resultant will be a more downstream resonances, right? So all these informations, when you pull together, you will get your structure. Definitely you will get your structure. So you have this is one example. You do it multiple 1D and 2D experiments. You assign, this is how you assign it for protons and carbons. You need to zoom in, and this assignment is very important. So this assignment is not that easy. It takes time, it takes understanding. You start your early NMR life with single compound, and where you know the structure very well, where you know the stereochemistry of the structures very well, where you understand what can be the cis trans positioning, where you understand what can be DL rotations, you understand the functional groups, you understand everything. Then you try to find it out through the NMR spectra. So once you get the NMR spectra and you correlate with your sample, then you see the similarity, and that is how your understanding of reading an NMR spectra keeps on developing. So a quick like suggestion maybe, when I started doing NMR, what I used to do is, I used to take a simple compound, as I said, the similar method, but I never used to reveal, or I never used to know what particular compound I'm using it. I used to try to get information from NMR, predict a model. Once I predict the structure, then I see how close I have been with my actual sample. right? So once it is like a jigsaw puzzle, you have a lot of zigzags there, here and there, bits and pieces are there, you pull together to get the entire structure. And that is really very interesting job. It's like it's very challenging at the same time. The more you do it, it's more fun. So we start with simple compounds, develop, and then go for complex and more number of compounds. And when you know that you have quite a bit of understanding, then it becomes more like a easy. By using the spectra only, you get a very very brief idea or a preliminary idea that what this structure can be. And then based on that, these days there are a lot of softwares which can also give you that model. So this is, you can see here, so this is through space. Like if I talk here, this is like 2 angstrom, 2.2 angstrom. So this a spatial orientation, the structural through the bond length orientation, the spin coupling, everything will give you the information to get into this final structure here. And that is why till date NMR is almost, you can say, the final instrument or the final modality through which a structural elucidation is always done. Be it any other modalities, but with NMR you get all the informations. I understand the number of experiments are more, you have to do this experiments, and then you have to understand all the different informations. But apart from that also, when you get the final structure, it will be in its best form, and it will be the actual structure of your sample. That is more important. You do not have chances of getting a false structure, or you do not have a loophole where you are not having much of the information is missing. You do not have the situation. So if you follow the protocol, if you dilute it, so the first thing is whatever sample you're using it, dilute it in a NMR compatible solvent. So NMR compatible solvent is all, it can be deuterated water usually, where you do not have a separate peaks. You can also use other like CDCl3. So there also you do not have a proton, so those things you can use it, and then use a proper reference. Once you use a proper reference, you align it, and then do start with a proton, start with carbon, single 1D experiment, and then go to the first is COSY, that is correlation through to 3 or 4 bond length distance. Once you do it, then you go for total correlations with all the neighboring spins. You can see the correlations. Once you have done that, then go for the Overhauser effect. So see, with NOESY, ROSY, HOSY, you can do all the Overhauser effect, and then finally you can do a heteromolecular studies, and with the heteromolecular quantum correlations, you can finally come up with a structure like this. And then you say, okay, this is my model. So these are some of the references. I'll be sharing it with you. And so this references, these are some of the textbooks you can refer, and these textbooks are really very extensive. If you want to have more in-depth information about NMR, you need to understand a lot of basic things, as well as the complexity in studying or in performing an NMR sample, or performing not only the experiment but also to understand the spectra. But I believe as and when you understand the gross idea, next time when you start with your NMR experiment, you will keep this small informations in your mind, and eventually you will land up in successfully elucidating the structure.